Hi, my name is Victoria Johnston, and today I'll be talking to you about a research experiment that we did at Bio 210 for the genetics lab. So the whole purpose of this experiment was to identify microbes for Superfund sites. Specifically, my Superfund site was Vincent, Alabama, and multiple microbes were identified to have been beneficial for plant growth and protecting against heavy metals. A little bit about Vincent, Alabama and why this is a Superfund site is because Vincent, Alabama is the location of a plating company that has been in production since 1956, but was determined a Superfund site officially in 2012. Wastewater that was generated from the Alabama Plating Company contained high levels of heavy metals including cadmium, chromium, nickel, and zinc. So below on the left corner is the methodology for this experiment. We began by getting a sample collection from our TA, which was Banuk Mohan, and we pulled apart leaves and roots and put them in a test tube, and then we put a PBS buffer. Then we created LB media plates that were either 100% or 1%, and then we got the cultures of the bacteria, put them in there. Then once a pure culture was formed, which was called the streaking plates, we ended up getting a bunch of the bacteria that grew in certain colonies, and we put them in another test tube, which we ended up doing DNA isolation, which was lysing it, and then we ended up getting the microbial DNA from that test, and then we ran it through another test, which was, it's a 16.5R DNA. It's an amplification and PCR production cleanup, and so once the PCR was ran, then we were able to get the actual Sanger sequencing and analyze the DNA and determine which microbe it specifically was. So above here are 4 pictures. The first on the left is sample 1 from site 3. That is the 100% media LB plate. The second picture is also from sample 1. It's the 1% media LB plate. The third picture is sample 2, and this was the 100% LB media plate. And the last picture was the 1% LB media plate. Those are pictures of once the bacteria grew for a week, we were able to take a picture and determine how many colonies were formed. So once the whole process went, we ended up getting the DNA sequencing. We were able to identify microbes, and the first microbe that was identified from sample 1 from the 100% LB media plate was Cyrtobacterium herbarium. And we used the NCBI, which stands for the National Center for Biotechnology Information. We used this website to then put the DNA sequence into there, and then we would get a percent identity back from previous samples that have been put in there, so it could show a more accurate representation of which microbe was identified. So for the Cyrtobacterium herbarium, it was 96.75% accurate, and the role with heavy metals is that it provided a resistance of aluminum and drought stress. The second microbe identified was Pantoia vagans. This is from sample 1 and the 1% LB media plate. This had an identity percentage of 97.55. And the role with heavy metals is that allowed lower arsenic uptake inside the plant. The third was Pseudomonas montillae. This was the second sample from 100% LB media plate. This had an identity percentage of 97.80%. This restores bacterial community in cadmium contaminated soil. And the last was Pseudomonas vancouverensis. This is from sample 2, LB media plate 1%. And the identity percent from the NCBI was 93.52. And this isolated heavy metal concentrations of mercury, copper, nickel, and cadmium. So as stated previously, Vincent, Alabama was a Superfund site that contained high levels of cadmium, chromium, nickel, and zinc. And all 4 of these had something to do with one of the high levels of metals that was also stated previously for Vincent, Alabama. So in the beginning, we had to form a hypothesis, and my hypothesis was if the plants were thriving in heavy metal concentrated soil, then there would be a positive correlation between the microbes, plant, and heavy metals. The microbes that were identified all had a positive impact on the plant that helped them survive in the levels of most heavy metals. Most microbes lower the concentration of heavy metals located inside of the plant. Now, these microbes can be very helpful to further understand environmental challenges that many plants can undergo, and it can also help us figure out how to combat these problems that these plants face. So the conclusion from my experiment was that the microbes identified during this experiment all benefited the plants from the effects of heavy metal concentrations. And the last part of this will be the acknowledgement. I want to thank UAB for allowing us to be able to take this 210 lab genetics lab, as well as the help of Banuk Mohan, which was our TA, and Shahid Mukhtar, which was our genetics lecture professor and the overall coordinator for all the genetics lab. And below are the acknowledgements for all of the roles of heavy metals that these microbes helped with.